Okay, so I just started the recording, and I guess we can go ahead and get rolling. So if you're just joining us, then I wanted to tell you that I'm sharing my screen now. This Q&A has been activated, so go ahead and start to either populate this with questions or look at the questions that have already been asked and start to say, yeah, I would like to see that one, or no, I'd like to not see that one. And we'll kind of get rolling here. So the other thing, if people can just kind of keep me apprised of what's happening in the chat, go ahead and feel free to interrupt and tell me, oh, this is going on in the chat, or someone said this in the chat, and that way I can respond to those things as well. So for now, though, if you don't have anything to say, go ahead and mute your mic. That way, not all of us are hearing what you're saying, and probably go ahead and turn off your video too, as that will help streaming across the entire time. Okay. All right. So let me just check and make sure that everything's going fine here. Okay. So everything's good. All right. So let's go ahead and get rolling. And looking at this, so the first question is practice exam short answer. So let's go ahead and look at that. I am going to go ahead and pull this up. I can write on this. I do have the practice exam pulled up as well. So let's just scroll down here and look at this. Okay, so the question says this: low blood glucose is detected by the pancreas. That subsequently releases glucagon, a hormone that has widespread effects, including the activation of gluconeogenesis in the liver. Given this information, predict the effects of gluconeogenesis activation on the following processes in the liver. Okay, so the whole idea here is that essentially we have gluconeogenesis running. So if you were to think about it in this way, we could write it out this way: pyruvate, okay, is being converted to, and we'll just take it as one long arrow, being converted to glucose. Okay, and of course we talk about enzymes that are available in that pathway, 4 of which we spent our time thinking about, and those are in those bypass reactions. Okay, we know that. And again, this is just generally speaking, we know that 6 ATP are going to be needed to do this. Okay. And we also know that you're going to need NADH to do this as well. Okay. All right, so we have this going on. So it says low blood glucose is detected. Okay, so given this information, predict the effect of gluconeogenesis activation in the following processes. Okay, so this is going on. Now it says A, rate of malate production in the mitochondria, right? So this is the reason that I drew it out this way, because remember, we have to have something, just like we did with cytosol, not cytosol, but glycolysis. We have to have something that will recycle, in this case, NADH to the cytosol and not NAD+. So with glycolysis, we looked at 3 different ways for the cell to recycle NAD+, right? Lactate fermentation. We talked about the malate aspartate shuttle and the glycerol 3-phosphate shuttle. All 3 of those recycle NAD+ back to the cytosol. Okay. In this case, though, we have to have something going like this. Okay, we have to have NAD+ recycle back to NADH, and one of the ways that that's going to be done is through the malate aspartate shuttle. Okay, so we expect then production of malate in the mitochondria, because that's really where it's going to start. We expect that to go up, okay, because it will go ahead and start to make the malate in order to then make, in order to ship electrons out into the cytosol, okay, and it's going to make malate from oxaloacetate. And malate will then be shipped out into the cytosol, carrying the electrons from NADH. Malate will be converted back to oxaloacetate. And in that case, what's happening here, I guess I can write this down instead of just explaining it. What will happen here is that we'll have malate converted to oxaloacetate. Okay, and that's going to then recycle that NADH. Okay, the electrons that are on malate will be given to the NAD plus, making the NADH. Okay, now interestingly, we know this, that the oxaloacetate will then be converted to pyruvate. Okay, and that will be done through PepCK. Okay, all right, so that's going to go up. All right, and this, by the way, I didn't draw this, but this malate came from, this is from the mito. I won't draw it out here, but it's from the mitochondria. Okay, so essentially the malate aspartate shuttle is running in reverse of what we talked about in class. Okay, all right, now B, glucose export out of the liver cell is going to increase, of course. Okay, so in this case, we have glucose being produced. That's going to be then exported out of the liver cell in order to then supply the blood with the needed glucose. Okay, and then finally, C, predict the effects that a cytosolic malate dehydrogenase inhibitor would have on the rate of glucose production in the liver. Okay, so let's do this. This enzyme right here is malate dehydrogenase. That's the enzyme that's carrying out this process right there, right? If that is inhibited, so what we're saying is that this can't occur, then we can't get the production of NADH, which means that this can't occur, which means that our rate of glucose production in liver will go down, okay? And of course, part D is just explain why, and C, and the reason is because we're not producing the needed NADH. And something else, we're not producing the needed oxaloacetate either. Okay, so both of those things are affected by this mutation, or not mutation, I'm sorry, but by this inhibitor. Okay, all right, so hopefully that helps you. And again, just to reiterate what I had said in the announcements of last week, when I said, well, you know, what do you want and what should you know? These are the types of things that you'll need to be able to do on the exam. You'll need to know these pathways and these shuttles well enough to be able to connect them and understand, well, what if I did this to the pathway? What would that do to this other pathway? Okay, so that's what we're going for in this. Okay, all right. And again, while I'm running this, don't hesitate to go ahead and interrupt me and you know, essentially on Microsoft Teams and say, I've got a question about that. Okay, and if you have something on chat, then let me know as well. Okay, so go ahead and feel free to speak out. By the way, it doesn't look like we have a lot of people going on this. Go ahead and start putting questions on here and saying, okay, that's a question I want him to do and that kind of thing, okay? And I'll try and get through these as many as possible, okay? I'm gonna have to leave a little bit earlier today, about 1:45-ish. I've got a graduation thing going on at CSU that I gotta be at. So, and they want me there a little bit earlier than I thought they did, okay? All right, so the next one, the bonus question on the practice exam, specifically the first one. Okay, let's go ahead and look at the practice exam. Bonus question. Okay, so this guy right here is what we're thinking about here. And it says this: there's a reciprocal regulation of glycolytic and gluconeogenic reactions. Which one of the following statements about this regulation is correct? Okay, and the individual says specifically the first one, F26BP activates PFK1. Okay, let me see here. Let's see if I can't. I'm going to read through these. So this is all insulin and glucagon signaling, which I think is another question. Let me go back to this. Could you walk us through the way insulin and glucagon affect PFK1, PFK2, or FBPase1, please? Okay, so let's do this one. And by doing that, then we'll get to that bonus question. Okay, so let's do that. And let's come here and go to this, get a blank sheet, and think about how insulin and glucagon are going to affect how glycolysis and gluconeogenesis are occurring in the liver, okay? And, you know, something to think about with this whole process is just the big picture, right? What is insulin telling the liver cell to do, and what is glucagon telling the liver what to do. And that's, if you can start there, that you have to, and then you kind of work your way backwards. I think that's a good way to do it. Insulin is a signal from the pancreas that blood sugar is high, and the liver, along with other tissues, needs to start to absorb glucose out of the blood. Okay. Compare that to glucagon. Glucagon is the opposite signal. It's a signal from the pancreas essentially to the liver that says glucose resources are low in the blood, and the liver should then act to export glucose. Okay, so it's going to undertake gluconeogenesis, and we didn't talk about this in class, but we also see that glycogen, which is a storage molecule of glucose, will be broken down. Okay, so that's big picture here. All right, and so when we do this, let's try and do it this way. Okay, let's just think about it this way. We're going to either make glucose, okay, and you need some source of pyruvate to do this. Or we're going to go in the opposite direction. I better leave myself quite a bit of room. We're going to see glucose being broken down, okay, to 2-pyruvate. Now, coming back up here, I said you'd have to have a source of this. Let's just say our source is like 2 amino acids or something like that. Okay, something along those lines, just to give you the idea that, oh yeah, that's right, we have to have something that makes the pyruvate, and amino acids would do that, lactate would do that as well. Okay, but what we have up here then, this would be gluconeogenesis, and what we'd have down here, this would be glycolysis. Okay, and essentially the direction of these arrows, you can think about it this way. If we're looking over here at gluconeogenesis, this is making glucose to be out of the liver cell, okay? And if you think about glycolysis, this would be essentially using glucose so that more glucose can be absorbed by the liver cell, okay? And so we're going to see this, that again, oops, we're going to see that insulin is going to positively regulate that, whereas glucagon, that little thing is in the way, there we go, is going to positively regulate that. Okay, now in the middle of this, okay, and kind of central to all of this is going to be this reaction where we have essentially PFK1, okay, and so let's just put here PFK1, the enzyme. And PFK1, let's see here, I'm trying to think about what's the best way to draw this out here. PFK1 is essentially going to be doing this, and we have, let me draw it down here, PFK1, and we have FBPase1 here. And what PFK1 is essentially going to do is it's going to take, as you, I'm sure all remember, or hopefully do, it's going to take this fructose 6-phosphate, I need to get my abbreviations right, fructose 6-phosphate, and it's going to make from that fructose 1,6-bisphosphate. Okay, and that's what PFK1 is going to do. Okay, let me go ahead and use a different color up here, try and help us out. This is going to be FBPase 1, and what it's going to do is it's going to do the opposite. Fructose 1,6-bisphosphate is going to be converted to fructose 6-phosphate. Okay, right now, where this would occur, right, in terms of the pathway, this would be PFK1 would be the third step. Okay, FBPase1 would be like the eighth, I think the 8th step. in the gluconeogenesis pathway, okay? But the arrows, right, the way they're going, either this direction, okay, or this direction, kind of tell you what pathway that particular enzyme is involved in. Okay, now here's central to all of this is going to be this molecule. Trying to figure out what color to use. Central to all of this is going to be this molecule, fructose 2,6-bisphosphate. Okay, fructose 2,6-bisphosphate is not fructose 1,6-bisphosphate. It's very, very similar, but it's different by the placement of where the phosphate is on the second carbon of fructose rather than the first. Okay, it's not a part of the pathways, not a part of gluconeogenesis, nor is it a part of glycolysis. Okay, and so what we see here is that fructose 2,6-bisphosphate is going to activate PFK1 while at the same time inhibit FBPase1. Okay, so if fructose 2,6-bisphosphate is present, then you're going to run, or the liver is going to run glycolysis and not gluconeogenesis. Okay. In fact, we could even draw this even more pronounced here. We could just say this, stop pathway, right? Whereas this one continues this pathway, right? Gives a big plus and really opens up the flux, if you will, for that pathway, okay? Right, so that's what's going on here. Now, here's where we're getting to this idea of glucagon and insulin. This molecule here, fructose 2,6-bisphosphate, can also be broken down, okay? It can be made or broken down, so let's go this way. It can be broken down to fructose 6-phosphate, or it can be made back into fructose 2,6-bisphosphate. And how does that happen? Well, that happens by this enzyme, this bifunctional enzyme, if you remember from class, where we have FBPase 2 or PFK2. That's real small writing, so let me zoom in a little bit here. Okay, and I'll make that a little bit nicer. Hopefully I can make that a little bit nicer. It's FBPase 2. Okay, and FBPase 2 does this. It breaks down fructose 2,6-bisphosphate. Okay, and PFK2 essentially makes fructose 2,6-bisphosphate. Okay, all right. Now, and that's all done in the same enzyme. Okay, and what is happening here then is this. Let's come back to our signals here. Is that insulin is actually going to activate this. Okay, whereas glucagon, on the other hand, is going to activate this. Okay, so glucagon is essentially acting to activate FBPase 2 that then breaks down fructose 2,6-bisphosphate, that then removes the inhibitor of FBPase 1 and then removes these red lines, essentially. Okay, whereas insulin activates PFK2 and causes the production of an activator of glycolysis, okay, activates PFK1 and it inhibits gluconeogenesis, okay? So that's essentially what's going on here. That's the big picture. Now, in class, these lines, right, we went through a little bit more detail on those lines. In particular, this one up here, we talked about how the glucagon will activate something called adenylate cyclase, and adenylate cyclase will make cyclic AMP, and cyclic AMP will then activate pKa, and then pKa will phosphorylate this, and when it phosphorylates this, then it activates FBPase2 and inhibits PFK2. Okay, insulin, through a variety of things, removes that phosphate. Okay, right? I won't draw all of that out. I'll let you kind of look back at your notes at that, but we did talk more details about those things, okay? Right, so hopefully that kind of diagram, it's a different diagram than what we did in class, okay? So now you have a couple of resources to try and pull from to try and hopefully figure out insulin and glucagon signaling. Now, let me go back to our practice exam because someone asked this question, the bonus question, so let's look at this. There's a reciprocal regulation of glycolytic and gluconeogenic reactions. Which one of the following statements about this regulation are correct? So we just kind of talked a little bit about that. But it says this F26BP activates PFK1. Yeah, that's true. So that's what I'm trying to illustrate right here. Okay, that's this whole thing. Okay, that's what's being illustrated there. Right? Now, let's go to the next question, see if it relates to that. PKA phosphorylates and activates the kinase domain of PFK2 FBPase2. Okay, so this gets into a little bit more of those details that I said, oh, we'll go back and look at your notes. Let's come back here. That question is related to this, right? Just a second, I have to remind myself of this. What is the question? PKA phosphorylates and activates the kinase domain, okay? All right, so glucagon signaling leads to the activation of this PKA, okay? And PKA is going to phosphorylate this, but it doesn't activate the kinase domain. So this PFK2 is what's called the kinase domain, okay? And FBPase2 is called the phosphatase domain. So PKA actually is going to phosphorylate and activate the phosphatase domain. So that's why that's false, okay? Not the kinase domain. Okay, and therefore by activating the phosphatase domain, it then removes the inhibitor of gluconeogenesis. Okay. Right now, 3, cyclic AMP levels in the liver cell rise due to glucagon signaling, activating PKA, whose actions can result in a decrease in fructose 2,6-bisphosphate. Okay, so this whole question is demanding that I do a little bit more with this. All right, so let's do a little bit more with that, okay? And so what we're going to say is this: glucagon is going to activate an enzyme called, oh, well, I better make it a little bit bigger. It's an enzyme called adenylate cyclase. Okay, and adenylate cyclase is going to do this. It's going to take ATP and make cyclic AMP. Okay, cyclic AMP then is going to activate PKA. So this is going to now be active. So we'll put a little plus there. PKA is then going to do this. So we'll draw our bifunctional enzyme here. We'll put PFK2 in the top, the kinase domain, and we'll put FBPase2 on the bottom here. Okay, and it can exist in two forms. It can exist in the unphosphorylated form. Oops. I'm going to copy that, duplicate. Oops. Delete. There we go. Sorry. I thought that would be easy, but it's not going to be easy. So we'll just do it this way. Be easier to draw it. So, but it can exist in two forms. PFK2 in the dephosphorylated and in the phosphorylated form. Okay. And when we have it in the phosphorylated form, so we'll just put a phosphate here, right? That's going to get the sunbeams coming out of this, right? Right? And that means that this is inhibited and this is activated. Okay? Well, PKA is going to do this. Right? It's going to activate or phosphorylate that bifunctional enzyme. That's going to activate FBPase2, which is then going to do this. It's going to take fructose 2,6-bisphosphate and break it down to fructose 6-phosphate. Okay, now this can of course go back in this other direction, can become dephosphorylated, and insulin's going to do that. And we just kind of drew a bunch of arrows and just said it's pretty cool, but it's really complex. And so then there's this protein phosphatase that's going to do that. Okay, and when that happens, this gets the sunbeams, and that means that this is activated and this is inhibited. Okay. All right, so that fills in those little things. I'll come right back to this. I know some of you are still copying that down, but it fills in essentially right here and here. All right. And so then this, just to be clear here, this is going to take essentially fructose 6-phosphate and make fructose 2,6-bisphosphate. Okay, and then do those things that we've talked about on the slide right above this one. Okay, in terms of fructose 2,6-bisphosphate, what it's going to do is, and that's either activate glycolysis through PFK1 or inhibit gluconeogenesis through FBPase1. Okay. Right. The nomenclature on this is not fun. I just don't shoot the messenger, right? I didn't make it all up. Okay, but this is the nomenclature that we're stuck with in biochemistry. Okay. Now let me come back to this. Okay. Come back to this question number 3. Cyclic AMP levels in the liver cell rise due to glucagon signaling activating pKa, whose actions can result in a decrease of fructose 2,6-bisphosphate. Let's come here. That's true, right? So glucagon signaling leads to a rise in cyclic AMP, so these are going up. That's going to activate pKa, which is going to then phosphorylate this, which is then going to decrease levels of fructose 2,6-bisphosphate. Okay, all right. Okay, let's come back here. Insulin signaling results in the deactivation of protein phosphatase. Let's look here. No, so this is the protein phosphatase. Some of you are looking at it and saying, what are those things that he put in that box? Those are P's. There we go. How's that? Protein phosphatase is what that stands for. It's a pretty general name for an enzyme. Okay, but insulin is going to activate that, and then that's going to dephosphorylate PFK2, FBPase2, and that's going to then make fructose 2,6-bisphosphate, which is going to activate glycolysis, which means the liver will start to absorb glucose. Okay, right. Now, come back here. Right, last one, 5, phosphorylated PFK2, FBPase2 leads to an increased flux through glycolysis. No, it doesn't. So phosphorylated PFK2, FBPase2, that's this. Okay, is going to lead to a breakdown of fructose 2,6-bisphosphate. If we come up here, when we're breaking down fructose 2,6-phosphate, we're removing an inhibitor of gluconeogenesis and an activator of glycolysis. So we'd have a depression in glycolytic rate and an increase in gluconeogenic rate. Right? The question that we just read stated the opposite. Phosphorylated PFK2, FBPase2 leads to an increased flux through glycolysis. It'd be correct if it said decreased flux through glycolysis. Okay. Okay. So hopefully that answers some of those questions. So I think we got that one. Could you walk us through the way insulin and glucagon affect PFK, FBPase1? We just did that. We've done the practice short answer, or the practice exam short answer. Next one, what's the role of tryptophan and tyrosine in membrane proteins? Great question. Let's go ahead and jump to that real quick. Go here, get to a blank slide here. And what we see here is this. Okay, let's draw a membrane. Okay, and it doesn't really matter. We'll just call this the inside of the membrane, and we'll just call this the outside, right? Not of the membrane, but of the cell. Okay, and what we have here then, we have these proteins that are going to be embedded in this environment, right? It looks something like that. Obviously, that's the high resolution structure of this. But here's the thing, okay? We know that the membrane is actually really more something like along these lines, right? And draw just a couple of these. And draw it here too. These fatty acids, right? Or not these fatty acids, but these lipids, phospholipids. Okay? And we know that then this is polar. And we know that this is hydrophobic. Okay? Which means that when you start to look at how a membrane protein is embedded into the membrane, it has a large portion of it, we just draw it right here, right? A large portion of it that's going to be surrounded, okay, by the hydrophobics. And so we've talked about what that means in terms of these membrane proteins, okay? But at the same time, there's going to be this thin little layer right here, right there, that will be interacting with polar head groups, and there's going to be this thin little layer right here that's going to be interacting with the polar head groups. Okay, well, it turns out that when we have a tyrosine, okay, let me go ahead and do this. Draw it like this. Tyrosine or tryptophan, I'll just draw tyrosine, but tyrosine is itself and tryptophan are both amphipathic. All right, so let's just go ahead and draw tyrosine here. It has a side chain that looks something like this. Let's just draw it like this. Trying to keep my coloring scheme here. That's a tyrosine side chain. Not quite done yet, but that's a tyrosine side chain. Notice that it's got this aromatic ring that's very hydrophobic, but then it has this hydroxyl group. And of course, I just go like this. There we go. It has a hydroxyl group that is hydrophilic. Okay, and it turns out that these tyrosines and these tryptophans, because they're amphipathic, will be found right at the edges of our membrane proteins, okay? So you'll find a lot of them right where I've kind of drawn this. This is not really to scale, okay? But you'll find tyrosines and tryptophans right at the in the membrane protein, right where that polar-nonpolar interface is, because they're themselves amphipathic, and they have a really nice way of anchoring the protein into the membrane, okay? Okay, so hopefully that makes sense to you. Okay, I don't want to go there. Where is it? There it is. Okay. Again, feel free to interrupt me if I didn't fully answer your question, okay? All right, so how do PFK1, FBPase1 illustrate the principles of metabolic control, specifically in regards to energy charge or energy change, okay? Great question. Let's go ahead and do that. I'm gonna run out of room here, so I can always erase things though. Okay. All right. So, all right. Let's think about this, and let's draw it this way. Fructose 6-phosphate, fructose 1,6 bisphosphate. Okay, and then we'll draw up here glucose going that way. And then we'll have it continue on this way, and this is going to make eventually, right, pyruvate. Okay, and we're going to get a bunch of ATP out of this. So let's just say it's 4 ATP down here in this bottom portion of it. Okay, so that would be the energy generation phase. Okay, on the opposite side of this, let's use different colors here. You would have gluconeogenesis. Okay, starting at pyruvate going up. Okay, and really again it starts at amino acids or lactate. Okay, but in any case, this is going to require ATP to do this. Okay, and so we can then have some example of this, but let's then look at this step here. And let's look at this step here. This step here is catalyzed by the enzyme PFK1. Okay, and this step over here is catalyzed by the enzyme FBPase1. Right? And it turns out that when we look at this, okay, that this molecule, oh, some of you are saying he's done it wrong. Red check minus. I messed this up. Okay, it should have been like this. Should say for ADP, sorry, makes 4 ATP. Sorry about that. Okay, I probably said it right and wrote it wrong. All right, so in any case, what we see here is this. This molecule here will activate PFK1, okay? Meaning this, that if there's a lot of this in the cell, that means that there's a low, what we call low energy charge, or the energy status is low because there's been a lot of ATP hydrolysis, which has resulted then in a lot of ADP. That will feed back and activate PFK1 and essentially say that, okay, there's just not enough ATP production going on. And so it's going to then open up the glycolytic pathway, okay? This inhibits PFK1. Okay, so in that case, when there's a lot of ATP in the cell, that means energy status is fine or high, meaning there's not a lot of drain on the cell for ATP, okay, usage. In that case, then it's gonna, that molecule is going to essentially inhibit PFK1 and slow down glycolysis so that glucose is not being made, okay? Now, I can't use a really cool drawing here, but what we can say is this. AMP, I guess I should have used the red, but AMP will inhibit this. Okay. Inhibits this. Right now, AMP is essentially going to be an indication that there's been a lot of ATP hydrolysis, and meaning that ATP is being used, and it should then feed back and inhibit any pathway that would oppose glycolysis, and that would include gluconeogenesis. Okay, all right. So what this is illustrating in terms of energy charge or energy status in the cell is that ATP and ADP and AMP are indicators of what the cellular energy status is. Okay, if it's high, there's going to be lots of ATP and glycolysis rate should go down. Okay, if it's low, there's going to be lots of ADP and AMP, and therefore anabolic pathways like gluconeogenesis should slow down, and glycolytic pathways should speed up. Okay, all right. So hopefully that clarifies that question. There we go. Okay. We did the bonus question, 56 through 60 on the practice exam. All right, let's look at that. Is anyone, is there chat, anything going on in the chat here? Just in case I missed it. I'm always going between Zoom and MS Teams, so I can't remember where the chat is. There it is. All right. Okay, so will we be able to scratch paper on the exam to draw out these pathways? Yes, you will. I think we're always able to use scratch paper. Yep. Shouldn't ATP be created in an energy generation phase? Yeah, that was the red check minus I got. So hopefully I corrected that. Sorry about that. You can give me the angry face on that one, right? So does ATP increase gluconeogenesis then? So it doesn't. At least not at the step that we're looking at, okay, in terms of FBPase1 is not activated by ATP. Now, some of you might say, well, why not? And the answer is because. right? So I've said that several times in the Echo 360 recording, it just doesn't. So anyway, so just AMP inhibiting. Yep, that's right. That's right. Okay. All right. So let's go back to this. Let me go ahead and make this probably one of the last questions I'm going to look at here. Okay. And then we'll go from there. So 56 through 60 on the practice exam. Let's look at that. Okay. The conversion of 2 moles of glyceraldehyde 3-phosphate to 2 moles of pyruvate by the payoff phase. Payoff phase, another term for energy generation phase, in the glycolytic pathway resulting in net formation of blank. Okay, which of the following of those is right? Okay, so let's look at this real quickly. It's not too hard to look at in the short order. Essentially, what we're saying is this, right? We have G3P is the start of our pathway. So, or the start of the question for the pathway. So it's the energy generation phase. And that is going to, if you remember, our pathway is going to be converted to, maybe make this a little bit larger, 1,3-bisphosphoglycerate. Oops. Yeah, that was right, bisphosphoglycerate. And then that's going to be converted to 3-phosphoglycerate. Then that's going to be converted to 2-phosphoglycerate. And then that's going to be converted to pyruvate. Nope, I'm sorry. Phosphoenolpyruvate. And then pyruvate, sorry. Right? So that's the energy generation phase, or the payoff phase. Okay, and we know this also, that this step requires NAD plus and makes NADH. We know that this step is going to make ATP. And we know that this step is going to make ATP. Okay? Now, here's the fun thing, right? When we're talking about this in terms of one glucose molecule, we know that we have to put 2, 2, 2, 2, 2, 2, 2 in front of all of this, because it all happens twice. Because glucose is broken down into 2 of these molecules. Okay, so the whole thing happens twice. Now, what that means is this. That means 4 ATP, and that means 2 NADH. Okay, all right. So let's go back to our question now. All right, so it says in this, because it says the conversion of 2 moles, right, to 2 moles of pyruvate. Okay, so in this we have, the only right answer, I mean, we can just do it from there, is this one. Okay, 2 moles of NADH and 4 moles of ATP. Now, if you included, just so we are real clear, if we included the investment phase, or if you will, the energy investment phase, then your number of ATP goes down because you have to use 2 ATP at the top of the pathway, okay? And then it's 2 moles of NADH and 2 moles of ATP in that case, okay? So there's that. And then the other thing would be, well, what if this was lactate fermentation? Well, if it was lactate fermentation, then the NADH that's made is converted back to NAD+, and there's no net gain of NADH in that case. So, there's lots of different things that you can add on to this that you'd want to keep in mind in terms of the pathway, okay? Okay, let me do one more question, okay? I know I'm going to cut this a little bit short, but I got some other duties I got to perform today. So, but how are the differences in standard and free energy exhibited in ATP? Okay, so standard free energy, the ATP is like negative 30.5 kilojoules per mole. However, the actual free energy of ATP is more like negative 55 kilojoules per mole. And the reason is because in the cell, there's just more ATP than there is its breakdown products, ADP and inorganic phosphate. Whereas at standard conditions, those are equal to each other. ATP is equal in concentration to ADP in inorganic phosphate, okay? And because of that, then what you would say is your Q value is smaller under actual conditions, and that leads to more negative free energy change, okay? Let's do one more. Metabolism illustrating conservation of energy of redox reactions through oxidative phosphorylation. Okay, so I think that this is, I think it was like either a learning objective or a question on the study guides. So the main thing there is when we get to oxidative phosphorylation, so we've gotten to pyruvate. Pyruvate is going to be converted to acetyl-CoA, and then acetyl-CoA is going to feed to the citric acid cycle. Okay, and the citric acid cycle is going to then make an ADH. An ADH is going to be used by the electron transport chain to give up electrons to oxygen, essentially. Okay, and when it does that, it'll make what's called a proton gradient or an electrochemical gradient. Okay, that then is used to make ATP. So when we talk about the conservation of energy of redox reactions, moving electrons from NADH to oxygen is a very exergonic redox reaction that makes an electrochemical gradient that then is used to drive conformational changes in a protein to make ATP. Okay, all right. So anyway, okay, I have to go. Okay, so okay, let me answer this. Will the names and abbreviations be on the exam? Yes. So in fact, I have spaced them like every 10th question. I've put the abbreviations. Okay, so they will be there. Okay, all right. So I'm going to let you go. I've got other duties to do. So you're welcome. And for those of you who are saying thank you, and have a good rest of your day. And good luck on the exam tomorrow. You're going to do great, okay? And if you have other questions, go ahead and email me if this didn't get you there, okay? All right, so I'm going to take off. Take care, everyone, okay? And I got to stop this recording and make sure that we're okay, all right? All right.